Okay, so before I begin, I would like to thank the organizer for this opportunity to present this work and the audience for attending this talk. So in this talk, I will describe a meta-analysis to combine data from multiple tissues from the genotype tissues expression data to find more eQTL and eGenes. So before we begin, I would like to take a very quick review of what is the genotype tissue expression data, which is also known as the GTEx data. So this data has 44 tissues, and each tissues contain approximately 50 to 300 samples. And the same person can donate samples to many tissues. So in other words, the data in the tissues are not completely independent. So in this work, I look at approximately 15,000 genes that are expressed in all of these 44 tissues. So from the GTEx data, there's come this concept of eGene. So eGene are defined to be genes that have at least one SNP associated with their expression. This means that to find to find eGenes, you would have to first find the eQTLs. And so that's come the next question. So why do we care about eGenes? Because obviously you cannot answer this question. There's no point to do the work, right? So it turns out that eGenes are likely to be genes that cause diseases and vice versa. Also, from the GTEx data, there's also another observations that some of the tissues have very few samples. For example, if you look at brain tissues usually have less than 100 samples. So to identify eQTL and eGenes, we would need to somehow pool samples across tissues to increase statistical power. So then there's come another concept is that people realize that we can do eQTL studies by jointly analyzing the data across many tissues. And what people have found is that the same SNP can be an eQTL in similar tissues, but the SNP can behave very differently in unrelated tissues. So in any case, there has been a variety of methods that is used to model how a SNP behave across different tissues. And there are two notable methods so far. The first one uses Bayesian approach, and the second one uses meta-analysis and a linear mixed model. So both of these current methods are not very scalable because they can only be applied in data that have less than 20 tissues. In any case, both of these methods rely on the idea that there is some pattern among the eQTL studies that we can exploit. And this is the intuition for this talk. Also, for this talk, we will follow the meta-analysis approach because it is more scalable. Here, we apply this method to 44 tissues. So to illustrate the intuition for the talk, so suppose we condition on a gene expression and compute the correlation of the SNP effect sizes for the 44 tissues in the GTEx data. So here, this plot shows how different tissues correlate with each other. In this plot for this gene, so in this plot for this gene, the correlations are very high among the brain tissues. So this black box outline the brain tissues. I have omitted the labelings for the other tissues. So looking at this plot, suppose we want to find SNPs that are eQTL in at least one of the tissues for this particular genes. We can combine eQTL results from these brain tissues to increase statistical power. So to quickly recap, for the GTEx data, in each of the tissues, we have the effect size of the SNP for the gene. And some of the tissue have very low sample sizes. which means that we have very low power to test if a SNP is an eQTL. And this also means that we also have very low power to find eGenes. So then the idea is that we should combine outcomes from different tissues to gain power to find eQTL and eGenes. So meta-analysis is a method to combine outcomes from different experiments. So in our case, the experiments are the tissues. Because the SNP and gene relationship can vary across tissues, what we would like to do is apply random effects meta-analysis, because this allows the SNPs to have unique true effects on different on the same genes across different tissues. So let's consider a very simple example, just so that we can gain a very strong intuition on how this method works. So let's first assume that we have only one SNP X, and we are interested only in one gene G that is expressed in 3 tissues, A, B, and C. So for now, let's assume that each of the tissues, A, B, and C, do not share samples from the same individuals. So the goal is to test if X is an eQTL in at least one of the tissues, A, B, and C. If X is an eQTL, then G is also an eGene, because by definition, G has a SNP that is associated with its expression. So given this example, because we have the data from each of the tissues, we can compute the estimate the effect size of X in each of the tissues. So we use beta hat XA to denote the estimated effects of X in tissue A. So similarly, you have beta hat XB and beta hat XC. Because these beta hats are estimated from the data, we can also compute their variances. So to apply random effects meta-analysis, you would need 2 assumptions. The first assumption is that the estimated effects are the summation of 2 terms, the true effects and the noise. For the second assumption, you would have to assume that the true effects come from some distribution. And a very normal way to do this is to assume that the true effects come from some normal distribution, would mean mu. Independent with the same variance tau square. You would also assume that the noise is normally distributed with mean 0, independent, and their variances are the variances of the beta hats. So with these 2 assumptions, the estimated effects has this distribution. So now you have the distribution for the observed data. You can now conduct a hypothesis test where you would like to test if X does not have an effect in tissues A, B, and C. This is the same as testing if mu is equal to 0 and tau square is equal to 0. So before moving forward, if you look at this distribution for the estimated effects, you can see that the off diagonals are 0, which means that you are assuming these estimated effects are independent across all the tissues. But recall the observation that the SNP can have similar effects on the same gene across different tissues. So this was the plot that I showed previously. So this plot is very important. So I will keep this plot in the corner. So we know that the random effects meta-analysis does not consider the estimated effects to be dependent. So to make the random effects meta-analysis fit the observation from the data, we would like to add in a covariance term. So one very easy way to do this is that you can look at this distribution that the random effects model assumes, and make a very simple change by changing the identity matrix I into some matrix U, where U contain non-zero off diagonals, so that you can allow SNP X to have similar effects on the same gene in tissue A, B, and C. So with this new modification, you have a new distribution for the estimated effects. And observe that because you have off diagonal to be non-zero, now you are able to capture the fact that the estimated effects can be correlated across different tissues. So you would still have the same null hypothesis that X does not have an effect in tissue A, B, and C. So again, this null hypothesis is the same as testing if mu is equal to 0 and tau square is equal to 0. So how do we estimate this matrix U? So we in the paper, the way we estimate U is that we use SNPs, the other SNPs in the data, and we only look at SNP that have very low LD with the SNP X, and we treat these SNP like independent replication of X, so that we have two vectors. So here, beta hat 1A is the effects of the first SNP in tissue A, and this SNP has low LD with the SNP X. Similarly, you have beta hat 2A, beta hat 3A, and for the second vector, you have beta hat 1B, which is the effects of the first SNP that have low LD with X in tissue B. So given these two vectors, you can estimate the covariance of beta hat XA and beta hat XB, and that would be one of the entry in the matrix U. So because the gene tissues expression data has overlapping samples, because the same person can donate samples to many tissues, we have to somehow estimate the facts that these overlapping samples can somehow inflate our test statistic. So to estimate how the overlapping sample can affect our results, we can do a simulation. So in this simulation, we assume that the SNP have no effects in all the tissues. We also assume that there are no LD for the SNP, so that in the simulation, the data is very clean. We create two different data sets. In the first data set, we create data that have overlapping samples. In the second data set, we create data without overlapping samples. In both of these data sets, we apply the meta-analysis method with the covariance term, basically fitting the distribution that is shown there to the observed SNP effects. So when you apply the method on the two simulated data set, when you don't have any shared individuals, you can observe that the test statistic, which is the p-values for the hypothesis test, looks very much like a uniform distribution. However, when you have shared individual in the tissues, then the p-values for the likelihood ratio test is shifted towards 0. So you end up having more you end up having more positive tests than you wanted to. So given these two distributions, we can compute a correction factor, very similar to how we would compute a genomic control factor for GWAS study. So moving on to the results. So we apply this method to the GTEx data, and the goal is that we want to find eGenes in at least one of the tissues, and we compare three different methods. We compare the tissue by tissue methods. So tissue by tissue methods essentially say that for each tissue, you want to test if a gene is an eGene. This is equivalent to testing if there is at least one eQTL for this gene. The second method we compare against is the random effects meta-analysis. And then the third method is our method where we apply random effects meta-analysis with a covariance term. So this is the results. So by applying the random effects method with covariance term, we detected 3% more than the traditional random effects model, and 20% more than the tissue by tissues method. And the Venn diagram shows us how agreeable are the three methods. So looking at this Venn diagram, you would notice that there are some genes detected to be eGenes only by a specific methods. So in this slide, we look at some of the cases to understand why some eGenes are detected only by some certain methods. So our methods relies on the fact that a SNP needs to have similar effects on the same gene across tissues. So in the first case, you can see that the tissues don't show any correlation pattern for the SNP effects. This means that this data don't fit our assumption, and therefore the method cannot detect that this gene is an eGene. In the second case, the tissue do have very strong correlation pattern for the SNP effects. And so from the data, we can see that this data fits our assumption very well. And so we are able to detect that this gene is an eGene. In the last case, the tissue have very weak correlation pattern for the SNP effects. And in this case, it seems that the traditional random effects model seems to do the best. And so this is the end of the talk, and these are my the people that I work with, and my advisor Elazar Eskin is the first one on the right. I would also like to thank the Genomic Analysis Training Grant at UCLA for supporting me. And Ken Lang is the PI for this training grant. And I also have the code on GitHub if you're interested, and I will take questions now.
Any questions?
Okay, thank you for your interesting presentation. I have one question. Building your variance-covariance matrices, do you consider in the model to have interactions, or you're using only the, how to say, the variance of the major components and the effects of the model?
Yeah, so we don't really consider any. So if you look at this, distribution, basically there are only two. So I guess we don't really have any interaction effects because in this model we only have two unknowns, the mu and the tau square.
Yeah.
So if you do a likelihood ratio test, then for the alternative hypothesis, you just have to maximize the likelihood where you assume that mu is something non-zero and tau square is something non-zero. So we don't do any, so we don't have any interaction effects here.
Okay, thank you.
Yeah.
Are you going to test your method in some other data sets as well?
So we only have, well, at least I only have access to the GTEx data, so I haven't tried it on some other methods yet.
Seems to work well with
Right.
This particular data set.
Yeah.
Another question there.
So in the end, when you compare the three methods,
Right.
I think you are more charitable than I would have been, because it seems your method is correcting for a lot of stuff that the two previous methods have not. So maybe the things that the two other methods find are actually wrong.
So I'm not. So we are in the process of sort of double checking whether we find more false positive, or whether the other methods find more false positive. So I'm sort of working on it right now.
Okay. Any other questions? If not, let's thank our speaker again.